Welcome back to all the students. In today's video lecture, we are going to discuss about one of the most central topics of biology, and specifically in molecular biology, that is protein synthesis. We had already dropped a short video regarding the polypeptide chain and peptide bonds on your request. Let's get towards the topic, which is protein synthesis. So the title goes like protein synthesis from genes to proteins. Obviously, if you are aware about what genes are, here you can see there is a gene in the title slide itself, a gene. So if you are aware about what is gene, then you will definitely come to know that a gene produces an mRNA. Let's discuss gene a little bit in brief. So DNA is a large molecule. DNA is a very large molecule which is having millions and millions of nucleotides over it. And these millions of nucleotides are made up of nitrogen bases, phosphates, and hydrogen bonds, and all other things. So this huge long DNA molecule is having multiple genes over it. So what are genes? Genes are simply few sequences of nucleotides. Nucleotide 1, 2, 3, A, B, A, T, G, C, A, T, G, C. These sequences, there may be hundreds of nucleotides, there might be thousands of nucleotides in one gene. It's a variable. So there is a huge nucleotide sequence which we call a DNA, and over DNA there is a stretch, a small stretch, comparatively small stretch of nucleotide bases, to which we call a gene. And how do we specify a gene? Why this specific part is a gene? Like here, this specific part is a gene because this specific part only can produce a meaningful protein. Okay, this specific part only is able to produce an mRNA, which in turn can produce a meaningful protein. That's why we call only this part a gene and not any other part we call a gene. We do not call this part a gene. Okay, so let's move on. What is protein synthesis? So what is protein synthesis? Proteins are something very central to every living organisms. Understand this fact first, that proteins are something which actually works in our body. We always call that DNA is the master molecule, but DNA never gets out of the nucleus. So how does the work actually happening in our body, in every living organism? How it is happening when the DNA is not actually coming out of the nucleus? So what happens actually, the DNA is having all the information about our body, and every living organism in the organism's respective DNA is having all the information about that organism. When that organism cells will replicate, when exactly that organism will die, when exactly that organism will have a specific disease. Even all these information are written inside the DNA. Every information is actually written in the form of codes we call nucleotide bases. Everything is there in the DNA. Information is there in the DNA, but DNA does not gets out of the never gets out of the nucleus to work. So what exactly happens? DNA transfers the information to mRNA first. Yes, the DNA transfers the information to mRNA first, and then mRNA gets out of the nucleus. DNA is transferring the information to mRNA. This process is happening inside the nucleus, and then RNA is getting out of the nucleus. Once it gets out of the nucleus, then it gets into the cytoplasm. There an interesting thing happens, which we are going to discuss in the next slide. So let's talk about the central dogma of molecular biology. In some texts, in some literature, it is called central dogma of life. Okay, central dogma of molecular biology. Information flows in one direction. It's quite clear. It never gets other way around. It never gets from protein to mRNA to DNA. We can definitely break down into proteins into amino acids, and we can definitely come to know what mRNA is responsible for producing which type of protein. And in turn, if we come to know the mRNA sequence, we can definitely come to know the actual DNA sequence which was responsible for production of this protein. We can go other way around actually in lab. But in real life, can a protein make a DNA in our body? It never happens. Only DNA can make an mRNA, and this mRNA will in turn produce a protein. So this is a one-way flow, only one-way flow from DNA to RNA and from RNA to protein, which we had already discussed. That DNA is having all the information. It carries, actually transfers the information to mRNA, and that mRNA is producing the proteins. Then this principle proposed by Francis Crick, very big name you can say. Francis Crick is no simple person. He was a great molecular biologist, Francis Crick. And then the cornerstone of molecular biology. This actually, whenever Francis Crick gave this principle, he actually, when he discovered this protein synthesis process, it was a real breakthrough indeed. It was a breakthrough to biology, and it was a breakthrough to molecular biology at that time, because nobody knew exactly how this process was going on. Then the first step, let's talk about the very first step of protein synthesis. So by this time, I hope you are already knowing what is the first step of protein synthesis. The first step of protein synthesis is to transfer the information from DNA to RNA, the mRNA. mRNA stands for messenger RNA. M stands for messenger. So the formation of mRNA is the very first step in protein synthesis. How does it happen? Here it is. It is called the transcription. The name itself is very much sufficient to tell you people what's exactly going on. Trans, across. Scription, the information is going across from DNA to mRNA. Let's see the diagram first. Here in the diagram itself, you can clearly see a double helical structure. You already know what a double helical structure means. It is a DNA. It is representing a DNA. Two blue ribbons helically structured. Okay, this is DNA, and over that DNA there is an enzyme called RNA polymerase. Here, this large circular area, this large circle is called the RNA polymerase. RNA polymerase is responsible for joining the RNA nucleotides to each other. And as you can clearly see, this strand over here is acting as a guide for the process of transcription. This strand here, which is beneath, this strand is actually guiding how exactly the mRNA should be built. This is actually RNA polymerase. This RNA polymerase is joining together various nucleotide sequences of mRNA to one another. As you can see clearly here, the mRNA is getting produced, and slowly the RNA polymerase is moving in the forward direction. And in this way, slowly over this strand of DNA, mRNA is getting produced. So the formation of RNA is the very first step in protein synthesis, formation of RNA. Only one DNA strand acts as a template. Template means a guide. Template means a kind of an SOP, standard operating procedure. You can see standard operating procedure means a template, simple guide how exactly an RNA is supposed to be built, how exactly it is supposed to be synthesized. Only one DNA strand acts as it, because we know the fact that these nucleotide sequences are very specific. They are highly specific. If we change the strand of that same gene, if we change the strand, the whole DNA sequence will get changed. The whole mRNA sequence ultimately will get changed. And even if a single nucleotide base is changed, it's called a mutation. It will lead to certain diseases, and some faulty proteins may get produced. So we cannot take chances here. We have to know which strand is acting as a template strand. So here you can clearly see the strand which is beneath it, that is acting as a template strand. It cannot act as a this other strand which is here over here. It cannot act as a template strand for this protein. The whole protein structure will get distorted. It will get disturbed by the changed sequence of it. Then mRNA is synthesized complementary to DNA strand. It is exactly complementary to DNA strand. Complementary means, if here you can see, we'll use a pen here. If there is thymine in the DNA strand, so in front of thymine only adenine will be there on the RNA strand. Okay, adenine will be there in front of thymine. So in DNA, if there is thymine, so on the RNA there will be an adenine. So it is quite clear that it's a complementary thing in nature. The whole sequence goes complementary. There is no randomness, there is no confusion in between the pairing of the sequences. It's completely complementary to each other. Then in RNA, there is a difference between RNA and DNA. One of those differences is that in RNA, uracil replaces the thymine. Uracil replaces the thymine. We know the fact that DNA contains 4 nitrogen bases. Which are those 4 nitrogen bases? You can comment down below in the comment section as the video progresses. So we know that DNA contains four nitrogen bases. And what are those four nitrogen bases? One is adenine, the other one is thymine, then cytosine and guanine. These four are nitrogen bases present in the DNA. But are these same bases which are also present in the RNA? Are these the same bases? No. There is only one base which is changed, and that is thymine. Thymine, which is present in the DNA, it is not present in the RNA. So the question arises, if thymine is not there in the RNA, which other base is there in the RNA? So the answer is simple. There is uracil. Uracil, which is present in place of thymine in the RNA. Thymine is present in the DNA. Uracil is present in the RNA in place of thymine. This copying process is called as transcription, and it occurs in the nucleus. We had already discussed that transfer of information from DNA to mRNA, it is actually occurring in the nucleus. So transcription in action. Let's see what happens here. The movement of message. How exactly message is moving from DNA to RNA. Genetic information is copied from DNA to mRNA. Yes, it's clear. The mRNA then exits the nucleus and carries the coded message to cytoplasm. This is a very important step here. Once the mRNA is prepared and DNA had already transcribed its information over mRNA, now mRNA is leaving, it is exiting the nucleus. It is exiting the nucleus and getting inside the cytoplasm. What is the reason for mRNA to get inside the cytoplasm? Can you guess? If you knew what is the reason why mRNA is getting out of the nucleus, please you can write it in the comments if somebody knows. When mRNA is getting out of the nucleus, there is a very specific reason, very clear reason. Because the protein synthesis which occurs and protein synthesis mechanism where it operates on the ribosome, and mRNA will only get the ribosomes into the cytoplasm. So the whole protein synthesis process is not going to carried out inside the nucleus. Only one process of protein synthesis, the transfer of information from DNA to RNA, will occur in the nucleus, and rest of the processes of protein synthesis, they are going to occur inside the cytoplasm. And for those remaining processes steps, the mRNA is leaving, exiting the nucleus and getting inside the cytoplasm. In cytoplasm, ribosomes catches the mRNA. Yes, ribosomes were waiting for mRNA. Ribosomes were waiting for mRNA in the cytoplasm. As soon as the mRNA gets into the cytoplasm, ribosome catches the cytoplasm and protein synthesis starts. Let's see how exactly. So the codes which DNA had encoded inside the mRNA, they look like this. These all are the codes. All of these are codes. Nearly 64 codons. Okay. 3 nucleotide bases. Pay attention. It is very crucial here. 3 nucleotide bases in continuation are called one codon. Okay. Are called one codon. 3 set of nucleotide bases are called codons. Remember this. And one codon is exactly after another codon. They do not overlap over each other. It's not like that 1, 2, 3 ends here and 2, 3, 4 will start here like this. No, it never happens. One code is here, another code is here. They are completely separated from each other. So one codon is made up of 3 nucleotide bases. So it is called triplet codon. Why it is called triplet codon? Now you know exactly why it is called triplet codon, because each codon contains 3 nucleotide bases. Yes. And every nucleotide base, now again, this is an important step. Every nucleotide base, every 3 nucleotide base codons code for one specific amino acid. Now what are amino acids? Like DNA is made up of nucleotide bases, RNA is made up of nucleotide bases. In the very same way, proteins are made up of amino acids. So amino acids are what? Amino acids are building blocks of protein. Okay. So each codon codes for a specific amino acid. And which codon codes which amino acid? It is a full chart given here, which you can observe. Just you can pause the video and you can observe the complete chart where it is actually telling you exactly which codon is responsible for which amino acid. So there are in total 64 codons, code all 20 amino acids, additionally a stop codon. Now, this is a very breakthrough discovery actually done by an Indian scientist. Yes, and for this discovery, he also won a Nobel Prize of 1968. Dr. Hargobind Khurana, his name is Dr. Hargobind Khurana, Indian American biochemist he was, and he deciphered the genetic code written inside the mRNA, written inside the DNA, and which codon is responsible for which amino acid, providing codons a triplet of nucleotides. So he was a pioneer actually. He and his colleagues actually were pioneer who actually came to know that the codon, triplet codon, how exactly a triplet codon works to produce a meaningful protein. So they decoded the genetic code, confirmed the triplet nature of codons because previously it was not known that three continuous nucleotide bases, which is called a triplet codon, is actually responsible for a protein, for an amino acid in the protein. It was not known. There were many tries scientists have done previously, but no one was actually coming successful. But Dr. Hargobind Khurana and Nirenberg and Holly, these three were the scientists who actually discovered the triplet nature of codons. This is the step 2. As we had already discussed, the step 1 is to transfer the information from DNA to mRNA, which is called transcription. Now what we are doing, we are simply producing proteins from the information given by mRNA. Now we are practically making something which we intended to produce at first. We were actually started everything to produce a protein, and now we are producing the protein. That's why we are calling it a direct translation and not transcription too, because we are not transcribing information anymore here. We are simply directly producing a protein now from the information which we received from the DNA. Okay, so the second step of protein synthesis, it is translation. We are directly producing a protein. So in this process, role of tRNA, which is another type of RNA, we had already discussed the ribosome, and rRNA is present into the ribosome. So role of tRNA becomes very crucial. Role of tRNA is very crucial. How exactly role of tRNA becomes very crucial? As you can clearly see in the diagram, just observe the diagram here. You can clearly see there the yellow part here, this is a tRNA. This is a tRNA. The green part, which is here, it is an mRNA which we received from the nucleus. It is coming from the DNA. And here, the ribosome, which is actually producing a peptide bond, making a peptide bond between one amino acid and the other amino acid. In this way, producing a protein, producing a polypeptide chain. What tRNA is doing here? tRNA is actually carrying, as you can clearly see here, observe carefully, tRNA is carrying the amino acid. Yes, he is carrying the amino acid. So if you want to produce proteins, having tRNA is a compulsion. So the tRNA, exactly tRNA, why tRNA is so important? Because tRNA is carrying the amino acids, the building blocks of protein. So tRNA actually is a very key player in the second step of translation, when we are translating the information from nucleotides and producing a protein, a functional protein here. So tRNA carries an amino acid. So the ribosome is here, present here, and mRNA is actually here, and tRNA are carrying specific amino acids as per the information present on the mRNA. And how it is done? How exactly tRNA knows which amino acid is need to be added into the growing polypeptide chain? How exactly tRNA knows this? Because tRNAs are having anticodons here. As you can clearly see, anticodons are given here. These are the anticodons. In this case, AAG is the anticodon on the tRNA, and UUC, which is the codon present on the mRNA. So UUC on the mRNA and AAG on the tRNA, they get along like this, and they check exactly, tRNA checks exactly that this information is there. Yes, this information is there, and we need to add this specific amino acid to the growing polypeptide chain. And ribosome captures that tRNA, and ribosome takes that amino acid from tRNA and make it a part of polypeptide chain. So tRNA carries amino acid to the ribosome. Each tRNA has an anticodon region, as you can see in the diagram itself. So it can actually check which amino acid needs to be placed into the growing amino acid chain. Anticodon is complementary to the mRNA codon. It is complementary to mRNA codon. That is again a very important point here to know that mRNA is containing codons and tRNA is containing anticodons. So nature is not random. Nature is not playing random. It's playing specific with codons and anticodons. This decoding process is called as translation because we are decoding the message given from the DNA and coming from the DNA with the help of mRNA. We are decoding this message into proteins. Now, codon and anticodon pairing. mRNA codon, which are present on the mRNA codons, AUG, start codon, it is there. Codes for methionine, which is always the first amino acid in proteins. Then there is tRNA. tRNA anticodon, UAC, which binds to UAG, delivers methionine, a specific amino acid. Why it matters actually? Because it ensures the correct amino acid is selected. Because even a slight change, even the change of a single amino acid can totally change the scenario. It can produce a disease actually. If a wrong amino acid gets added to the polypeptide chain to the protein, it may differ into the conformation of the protein, ultimately making a default of protein, you can say. So ensure the correct amino acid is selected and added to the growing polypeptide chain. Amino acid are arranged in the proper sequence. That is again very important, obviously, to produce perfect proteins which can work perfectly in our body. So error in pairing, faulty proteins. It is a direct correlation. If there is an error in pairing, there is an error in the addition of amino acid, definitely the proteins are going to become faulty. Ribosome and protein assembly, as you can see clearly, this is a very colorful diagram, very helpful diagram. Here you can see tRNA is coming with a amino acid, and it is placing the amino acid here on the ribosome and making a pair here over here. Anticodons present on the tRNA and codons present onto the mRNA, they are getting paired, and afterwards when they are very much sure that yes, this is the perfect amino acid which is supposed to be a part of polypeptide chain, it gives the amino acid to the ribosome, and ribosome added it to the growing polypeptide chain. As you can see, this polypeptide chain is growing, which is ultimately going to become a protein. This whole big structure behind this is ribosome. The chain of mRNA is over here, and these three are tRNAs. Okay, so rRNA catalyzes the peptide bond rRNA, which is present inside the ribosomes. Yes, so ribosomes are also having rRNA, which catalyzes, which facilitates the bonding of one amino acid to the other amino acid, which we had already discussed in the short video we uploaded a few days ago. Polypeptide chains then. Polypeptide chains, amino acid links in sequence folds into functional protein. Ultimately, they give the final results. So if you like the content, if you like the presentation, please hit the subscribe, because we are coming up with more and more videos for your conceptual understanding. If you are having any doubt, any question, any query, even some improvisations in the presentations, you're most welcome. Just drop everything down into the comment box. If you want tailored, if you want specific notes for the board exams, you can say 10th board exams related to this topic, you can also get those notes from me. Just drop your question, just drop your queries into the comment box. I will tell you how exactly you can get those notes which are tailored for your board exams, not some random notes which you get, not random notes specifically for your board exams. You can get that also. Okay, so thank you everyone. We'll meet again into the next video lecture. Up until then, have a good day.